So this is a quick introduction to NextBio, or what is now called Illumina Correlation Engine. So what is Illumina Correlation Engine? It is, I believe, what we would call a biologist's dream to allow access to those that may not necessarily know R or other more sophisticated tools to get in and mine genomic data. So what they've done here is taken and processed all of the gene expression omnibus database, Array Express, a number of the GWAS databases, the tumor sequencing databases, and came up with gene lists, ranked gene lists, that allow you to put them all in a database, and then basically applied a correlation engine, hence the name, that allows you to look at relationships based upon the patterns in these different gene sets across an entire database. So you can go across species, you can go across technologies. It's all linked up based on genes. So I can look at the correlation between my, say, rat gene expression data in liver and GWAS findings in humans. Again, all linked up through genes. And so let's go through just a quick overview here. I'm going to start here. So you can enter into the correlation engine at many different levels. Oftentimes, I'm searching a gene, or I am looking for patterns. I'll upload my data, and there's a separate video on YouTube on how to upload your data so you can correlate it across all the studies in NextBio. It's on it should be on the playlist here. So let's just go into curated studies. And so you can search something, say, I'm a toxicologist by training. So let's look up one of our favorite chemicals, phenobarbital, because it has effects in the liver. And just search curated studies. And it's processing here, so just give it a second, and it will come up with a list of things. So this here happens. So there's a whole bunch of studies that used phenobarbital as a treatment, and they're all listed here. And so most of these can be found actually in the GEO database. So let's just look at this first one here, which actually came from an EPA group. So this is one study set that you could open up. You can open up any one of these, and this happened to be one of the treatments in this study. There are others. So this happens to be a study called Triazole Antifungal Toxicogenomics in Primary Hepatocytes. And if you want to look at the full study details, here is the description that's captured in the database. It's from this GEO ID here, this GEO series. Gives you all sorts of information about it. You can link out to the GEO data set, tells you about the authors, things along those lines. Sometimes they have an abstract in here. Tells you the number of biosets. Biosets are basically just gene lists, right? So you go here, and it shows you all of the different basically the comparisons that were pulled from this GEO series. So we have primary hepatocytes, phenobarbital versus vehicle, miclobutanil versus vehicle, so you get, and you can look at any one of these by pressing this window icon here, and it'll come up and it'll give you a list of differentially expressed genes, and we'll do that in a second. And then you can also look at the regulated genes across the data set. So each one of these little bars here represents one of the treatments. So if I mouse over them, that's phenobarbital, that's miclobutanil. Again, different concentrations, and it just shows you that CYP3A43, which happens to be I believe this was done in humans. This is a human microarray that was used to measure this. And if you look here, if we open this up, yes, it was an Affymetrix microarray. So it gives you all this sorts of information. So you can open it up and look at all of these things, and there's a tremendous amount of information. You can also look at active biogroups in the data set. It'll populate here in a second. Biogroups, so biosets are gene lists, and biogroups are basically what we think of as pathway sets or gene sets. So you can see the top differentially expressed gene set across across all the treatments in this GEO series is xenobiotic metabolism. Not surprisingly, that's commonly what you see in with gene expression studies in hepatocytes. So let's just go back to one of these biosets. Sorry, we'll pick one. So let's go back to our primary rat hepatocytes. So what I did is I clicked that little window icon, and it gave me a list of all of the genes that are differentially expressed, and it gave it to me by rank, and the rank is determined by the absolute value of the fold change. So if you want to sort by fold change, you can just press fold change up and down here. But now I'm going to demonstrate the real power of this tool, this data-driven analysis tool. So I have my gene list here, and imagine it's your gene list for whatever experiment you have. It could be gene expression, it could be GWAS. It's all mapped. It's all mapped to genes, and you provide a rank based upon some sort of statistic or effect size. Right? In this case, it's going to be effect size because we're looking at fold change. And so what you do is you simply go over here, you mouse over to this curated studies, and what it's going to do is going to give you a list of all the curated studies in the database, the truly massive database, which is the NextBio data set, and it'll come up here. And so not surprisingly, what you find is this treatment here, 1,000 micromolar phenobarbital versus vehicle, is correlated with one of the other treatments in its study set. But then, as you start going down here, you'll notice that it's enriched. This treatment is actually enriched in liver-enriched genes, genes that tend to be expressed in liver. This happened to be one of my data sets, and they tend to be downregulated. And this is probably because of some dedifferentiation that might be going on when you stress hepatocytes. And then you have other studies from other groups that are lining up with this. So these are HEPA-RG studies. HEPA-RG is a cell line that looks and acts very similarly to a hepatocyte. And not surprisingly, another chemical treatment in hepatocytes. This happens to be a PPAR alpha activator, not a phenobarbital-like chemical, is actually aligning very nicely to this treatment. So as you go down here, you can see there's a tremendous number of studies that have linkage, and if you open up any of these, you'll see the association. Here's another one, HEPA-RG cells with CAR knockout phenobarbital treated versus DMSO. So there's probably some non-CAR related signaling where your phenobarbital is affecting hepatocytes, and that's why you're getting some degree of overlap. So you get the basic idea. What's also quite powerful about this tool, as you can go through and you can look at a variety of different things. You can say, you want to take this gene list and look to see where it's expressed across the body, what it's most correlated with. And I already showed you this a little bit with the liver-enriched genes. It tends to be these genes that are differentially regulated tend to be expressed very highly in the liver, or enriched in the liver. In this case, green indicates that they're particularly downregulated. So the genes that are typically up in liver relative to other tissues are downregulated by this treatment. Also will tell you what associations you have with different diseases. So they've gone, the NextBio folks or the Illumina folks have gone through and curated what the gene expression data sets that they have from different diseases, and then they can relate them or map them to your analysis. So in this case, this happens to be liver cancer is one of the most strongly associated, not surprisingly. And so what you can do is you can open this up, you can click here. So once you've gone through the disease atlas, and it takes you back over to all the associations with your gene expression list and the diseases. And so it happens to be that this gene expression list is actually closely associated with this gene expression list here, which is liver from patients with very advanced hepatocellular carcinoma versus normal liver. Again, this goes along with this idea of this chemical treatment producing some degree of dedifferentiation in the hepatocyte, so you're getting loss of liver-like biology. And as you can see here, when you do this comparison, so here's the 1,000 micromolar phenobarbital treatment, here is the hepatocellular carcinoma treatment, gives you a number of genes that are up and down regulated, gives you the positive correlation, and a negative correlation gives you the overlap, and it tells you what those genes are. And you can actually export this image or export the data if you wanted to make an argument in, say, a manuscript. So that's disease atlas. Let's look at pharmacoatlas. Tells you what what chemical treatments produce similar signatures to yours. So these are all curated chemical treatments that are in the database. Turns out that there are a number of chemicals that have unclassified mechanisms that are associated positively with phenobarbital treatment. This is, I believe, let's just take a quick look at this. So this happens to be, it's a perlizidine alkaloid. These tend to be genotoxic agents, things that damage DNA. And so again, we're comparing this primary rat-hepatocyte to, in this case, to treated with 1,000 micromolar phenobarbital to these hepatocytes in another study, completely different study, someone else did, looking at this treatment with this chemical versus the control, and you can take a look at the overlap. So this pharmacoatlas is a way of basically doing connectivity mapping, if those of you are familiar with it, similar to what is done with the CMAP data set at the Broad. Finally, you have the knockout atlas here. This is the last of the data-driven curation. So what they've done is similar to the disease atlas and the pharmacoatlas. The Illumina folks have curated gene expression studies that have come from knockout studies, and so they'll tell you what the relationship is between the pattern produced by your treatment and the pattern produced by a knockout. And so when you knock out BAG3, apparently, it produces a positive correlation with your treatment, and we can quickly open this in this case. And so you can have a look here. So there is greater positive correlation than negative correlation, although it seems to be somewhat balanced here. But basically what it's telling you is if you knock out BAG3, you produce a gene expression pattern that is similar to your chemical treatment. This is actually quite useful for looking at things like transcription factors that may be driving your association. So when you knock it out and the gene expression actually is inverse to what you're anticipating. So this happens to be very powerful, and it's actually worked out quite nicely for me in looking at other treatments. This one is not showing as much as I would anticipate. And finally, the traditional pathway enrichment. So there's many different tools for doing pathway enrichment. This pathway enrichment tool works somewhat similar to a GSEA-based process. And so you can just click on here, and what it does, it takes the list and does pathway enrichment, and it has a bunch of different data domains. So you can, if you wanted to, you can look at just gene ontology. You could look at canonical pathways, just simply apply a filter. And what you'll see here is, not surprisingly, metabolism of xenobiotics by cytochrome P450 is the highest scoring one, and they tend to be upregulated. So there's, again, very powerful tool. But one of the other components that I wanted to highlight in this video is the ability to do meta-analyses. So let's get back here to curated studies. And say I wanted to create a signature for phenobarbital effects in liver. So what I can do is I can go through here, and hopefully I can identify a couple of phenobarbital treatments. If I cannot, I might have... We'll take Citgo. Citgo is another CAR activator, so we'll take that one. So what I'm doing here is I'm going through and I'm putting, see this little icon here? I'm putting these into the meta-analysis. Notice how I've clicked two of them now. If I went over here, there would be this study, and then there would be this study. And we'll find a couple more of them just for illustration purposes. Hopefully I can find some phenobarbital treatments. There's one there. There's another one there. So let's make a signature for phenobarbital treatment in liver. So what we have here are hepatocytes. So we have 3 different biosets. Hepatocytes cultured in normal liver donors treated 24 hours at sit-go. Rat hepatocytes treated with phenobarbital. This is the wrong organism listed here. Hepatocytes from female donors for 24 hours for phenobarbital, and then female donors 24 hours with a higher dose of phenobarbital. And say I just wanted a signature of upregulated genes, sort of a non-exclusive lack of, sort of in this case, a lack of specificity signature, because all I'm doing is looking at positive treatments. I don't have any negative treatments in here, but you can add negative treatments and then say, I want it to be absent in this treatment, right? So this is what it does, is basically it does a search for those genes that are upregulated in these 4 treatments consistently. And so you can go here to the gene result, just clicked here, and thank goodness this worked. So CYP2B6 happens to be a gene that is commonly upregulated by phenobarbital. It's the best documented gene, and not surprisingly, it shows up quite nicely here. CYP3A4 is also on that list, CYP2C8, things along those lines. So you see, it doesn't work every time, but again, it's the wisdom of the crowd effect, right? We have multiple treatments here, and what it's showing you is those that are most consistently upregulated. And there's different ways of scoring them. There's a specificity analysis and a score, and the specificity relates to the settings that you had in the setup. It can also tell you what biogroups are consistently upregulated or in your set. And so, not surprisingly, phenobarbital is known to induce P450s. All the treatments seem to have a strong effect on this interpro group for cytochrome P450s, and then, of course, genes involved in xenobiotic metabolism. Again, consistent with the upregulation of the CYPs. And you can filter just like you did previously. We showed you previously in the pathway analysis. And then, say you wanted to look at your signature, and so you created this signature, and you want to get a quick look at what other data sets out there in the database actually have this signature. And so you can go to bioset results, and I think you can only do this with maybe up to 50, maybe only 10. I can't remember now. And so what it does is it finds you the different treatments that have a strong relationship with your chemical. Not surprisingly, there is a short-term 8-hour treatment with phenobarbital from one of the studies that contained these 2 treatments here. But you can see that there are a number of other treatments that have very, very similar patterns of response. So I did want to highlight one last thing. So that was the meta-analysis tool. So that's how you can create signatures. And you can always, if you want to, you can export this and then re-import it as a signature that you can then use to score across the database. So you've now come up with a sort of a meta-analysis derived signature. Say you know something about all these treatments that all produce one pathology, and you have a whole bunch that don't. You can come in here and create a signature, save it, and you can also bookmark this. That's the other thing too. So you can, if you wanted to bookmark this analysis, you can simply hit bookmark here, and then you can basically save your analysis, put a whole bunch of description in here, save bookmark, and then the bookmarks are here, so you can always come back to it once you're done, which is really nice. Finally, I just want to have a quick look here. Let's just go back to this Trizol antifungals. Go here again. We'll go to the rat hepatocytes. And I want to just quick show you that you can actually do associations here with, I don't know how meaningful they are necessarily in many cases, but with other data types and other, you can look at other species. So say I wanted to look at this relationship with certain SNP GWAS studies. So I'm looking at primary red. So apparently there is an association with SNPs from, oh, interesting, serum bilirubin levels, which serum bilirubin levels are regulated in the liver. And turns out that, if I'm recalling correctly, phenobarbital has been used historically to upregulate UGTs to clear bilirubin. So this actually kind of worked out well. So there's a polymorphism in this SNP here, in the UGT gene. For those of you familiar, UGT is actually it's actually an alternatively spliced gene the UGT1, and so there's multiple starting exons and one ending exon, so you can jump different early exons and get to, and then link to the same basically last exon. I believe if you wanted to look at that, you can actually jump over to the genome browser. So it gives you the relationship between the genes and the SNPs. And so you found an association here, well, we have, with phenobarbital associated induction in liver and serum bilirubin levels. So this might be a linkage between your functional genomics and your static genomics. So there's NextBio. There's much more to be had, and if you dig deeper and you find something that you think is interesting, I hope you share it on YouTube.